Hi, in this short lecture we're going to cover the basics of cells. So there are two major categories of cells, and most of the cells on the planet can fall into one of these two categories. The categories are prokaryotic cells and eukaryotic cells. Prokaryotic cells are the simplest form. They're much smaller than eukaryotic cells, as you can see from this image here. Here's a prokaryotic cell. Prokaryotic cells include bacteria and their close relatives, the archaeans, whereas the eukaryotic cells are basically everything else, so plants, animals, fungi. There's some major differences between pro and eukaryotic cells. Eukaryotic cells are known as eukaryotic cells because they have what are referred to as membrane-bound organelles. They also have a membrane-bound nucleus, and within the nucleus, DNA is kept. In prokaryotic cells, on the other hand, the DNA floats freely within the cell, and it's not enclosed within a nucleus. So let's take a closer look at prokaryotic cells. They're much smaller, and they lack internal structures. They don't have a nucleus. So in this picture of a prokaryotic cell, you'll notice several things. First, the cell is covered in cilia. These are small hairs that allow the cell to move. There are also long tail-like structures which are referred to as flagellae, or multiple flagella. The cell is kept within a plasma membrane. The plasma membrane allows it to exchange materials with the outside, and a thickened cell wall gives the cell some structure. The DNA is normally found in a circular strand, and it floats within the cytosol or cytoplasm. Ribosomes, which are used to make proteins, float freely in the cytosol. Eukaryotic cells are much more complicated and have many more internal parts. The DNA is found within the nucleus, which is bound with membranes. Surrounding that, you have endoplasmic reticulum. Endoplasmic reticulum that has ribosomes attached to it is called rough endoplasmic reticulum, and that's used to produce proteins. On the other hand, smooth endoplasmic reticulum is used to produce lipids or fats. Within the cytoplasm float other organelles, mitochondria, which are found in both plants and animals. These are used to power the cellular metabolism. On the other hand, we also have things like a Golgi apparatus, which is basically like a packaging factory for the cell. Centrioles will become important when we talk about how cells divide, and like the prokaryotic cells, they can have flagella and cilia and other structures. This is just an idealized view of eukaryotic cells, and when you learn in your e-text about plant cells and animal cells, I want you to pay attention to the things that they have in common and the things that are different. So a plant cell, in addition to having mitochondria, has another special organelle, which is referred to as a chloroplast. The chloroplast has stacks of green membranes, which are called thylakoid membrane, and it's used to create a differential in the amount of hydrogen ions within the chloroplast, and that drives a very important chemical reaction, which is referred to as photosynthesis. That's when the plant takes carbon dioxide and turns it into sugars. In addition to a chloroplast, plants have other structures that are different. A cell wall, which is generally supported with cellulose, and then there are little structures that allow the cell membranes between plant cells to interact with each other. Plants also have a large membrane sac, which is referred to as a central vacuole. It's used to store things like starch. Cells were discovered quite a long time ago, in 1665, and accumulation of evidence has led to what we refer to as the cell theory. And that is simply that all living things are created out of cells, and all cells come from previously existing cells. We can use scanning electron microscopes to help us to explore more about cells. And one thing that's interesting is when you look at the surface of a cell, it's generally not smooth. It has all sorts of structures sticking out from it. This is a picture of a white blood cell in your blood, and all of those little projections are actually proteins that are involved in your immune system. So on the membrane of a cell, membranes consist of multiple repeating units. These little things that have got circles with two little tails are called phospholipids. They have one part of them, the phospho part, which is represented here by a yellow ball, and that actually can dissolve in water, whereas the lipid part, the fat part, is repelled from water. So it's basically like a little fatty layer across each cell. In order to allow materials through the membrane, there are all sorts of transmembrane proteins. Some of them are involved in transportation. Others are involved in cell signaling. That's how your cells can communicate with each other. Some are involved in enzymatic activity, where they help to make chemical reactions occur. Other proteins help cells to join with each other, and then some are involved in cell-cell recognition. So you might have heard about somebody who had a transplant, for example, and they needed to make sure that the tissues were compatible. Your body knows whether or not a tissue is compatible using that cell-cell recognition. So let's look at a few of the important parts within the cell. The nucleus. This is bordered by a double membrane, and within the nucleus you have chromatin. Chromatin is DNA plus proteins. The endoplasmic reticulum produces all sorts of molecules and has both rough endoplasmic reticulum that contains ribosomes and smooth endoplasmic reticulum which does not. The rough ER produces proteins and new membrane. Ribosomes are very important because they build all a cell's proteins. We'll cover this in more detail in later lectures. Basically what happens is in the nucleus, DNA gets rewritten into an mRNA form. The mRNA leaves the nucleus, and when it's in the cytoplasm, ribosomes attach to the mRNA and read that strand of RNA to figure out which sequence of amino acids to make a protein. So the RER, or the rough endoplasmic reticulum, is important for producing proteins, and the smooth endoplasmic reticulum can actually be used to make lipids. And what's kind of cool about the structure is the ribosomes on the outside produce proteins on the inside of the endoplasmic reticulum, and they can get packaged and then transported inside of little vesicles. Those little vesicles can then be taken to the Golgi apparatus, which then processes the proteins in more detail and can make transport vesicles that then are used to excrete compounds outside of the cell. So for example, in your digestive system, you need to secrete compounds into the lumen of your digestive system. So the Golgi apparatus works in partnership with the endoplasmic reticulum. Vacuoles in plants can be used for different things. Storage and defensive compounds. Think about where do essential oils come from. They're stored in central vacuoles. In protists, which are not plants, but they're single-celled eukaryotic organisms, they can use vacuoles to help them to move themselves. They basically will contract the vacuoles and squirt liquid out of them, and that allows them to move with a sort of a jet propulsion. So the endomembranal system has rough ER, transport vesicles, Golgi apparatus, secretory vesicles, and a lot of these proteins that are made have to be kept inside of membranes. The reason for this is that many of the enzymes in your body are made out of protein, and if those enzymes were floating around, they would be working on things that you wouldn't want. It would be the equivalent of not carrying bleach in a bleach jar. You want to keep your bleach in the bleach jar with the lid on so you don't accidentally bleach your car or your bed, but you keep the bleach for the toilet or your laundry, and that's basically the same principle here. Chloroplasts, these are the sites of photosynthesis, and mitochondria are the sites of cellular respiration. We'll cover these in a lot more detail when we hit those chapters. We're going to spend a little bit more time talking about the membrane structure and how compounds move through membranes. I do want you to pay attention to the fact that phospholipids are the compounds that make membranes, and that most membranes have proteins embedded in them that allow them to do different things.